Hello, my name is Brian Thompson, and I am a fifth-year PhD student at the Yale School of Public Health in the Department of Environmental Health Sciences. As a PhD student in the School of Public Health, I've had the opportunity to learn how to think about my research from a unique perspective. I'm constantly challenged to think about how my research impacts patient outcomes, their lives, and their standard of living. This challenge has ensured that my research, which is focused on advancing our understanding of the mechanisms of disease at the molecular level, is grounded in the patient experience. My belief that vision is a critical component of the human experience motivated me to study the eye. As I began developing my PhD research program, I realized that much about one disease in particular, called microphthalmia, remained to be elucidated. Microphthalmia is a disease in which one or both of the eyes have failed to develop properly, and so they appear small. In patients with severe microphthalmia, vision is compromised in the affected eyes. This leads to microphthalmia being a major cause of childhood blindness cases, and worldwide, childhood blindness costs upwards of $270 billion annually. Current treatments can only address the cosmetic effects of microphthalmia, and tragically, they fail to restore vision. Therefore, it is essential that we understand more about what causes microphthalmia, as this may allow us to develop strategies to prevent it from occurring in the first place. Roughly 80% of microphthalmia cases can be attributed to genetic mutations, which means that upwards of 20% of cases may be caused by exposures to environmental factors. Indeed, much epidemiological research has associated prenatal exposure to maternal infections, alcohol, pharmaceuticals, and other environmental factors with microphthalmia. Critically, though, much about the mechanisms by which these exposures can impair eye development and cause microphthalmia remains to be known. My PhD research has focused on one potential mechanism by which environmental factors could cause microphthalmia, namely oxidative stress. Oxidative stress is simply an imbalance between good antioxidants and bad antioxidants in favor of the oxidants. By using a genetically engineered mouse model, I was able to induce oxidative stress in only the developing mouse eye. Amazingly, these mice developed severe microphthalmia in both eyes that was characterized by morphological changes to the cornea, lens, and retina, with the lens being most severely affected. I then focused my investigations on the lens with a next-generation sequencing technique called RNA sequencing. My RNA sequencing of only the developing lens tissue suggested that oxidative stress was impairing lens development by hindering the function of a transcription factor that is required for normal lens development, called PAX6. My data is the first to suggest a mechanistic role for oxidative stress in regulating eye development through the alteration of the function of PAX6, and thus adds to a built-in body of evidence which suggests that aberrant oxidative stress can cause severe birth defects. My data adds further support to previous reports that the function of members of the PAX protein family can be impaired by oxidative stress. This finding can have immense implications for other diseases in which PAX proteins are implicated. It is hoped that by developing a better understanding of how environmental factors can cause microphthalmia, we can develop therapeutics and strategies to prevent the disease from occurring in the first place. These strategies could even be as simple as providing pregnant people with guidance on which chemicals to avoid.